I wanted to talk today about why are dose constraints from the more well-known modality, external beam radiotherapy, not directly transferable to radiopharmaceutical therapy. Because it's, I think, an important question. Pardon me. As an outline, I'd first like to start by introducing what it comes down to is differences in dose distributions between RPT and EBRT, or external beam radiotherapy, which kind of drive differences in the radiobiology, which is my area of current research interest. And so I thought I'd start by giving an overview of kind of the basics of external beam radiobiology. And then, so that if we understand that, we can understand how RPT radiobiology is different. And that will kind of segue into giving an understanding of some of the research that I've been working on over the past few years on trying to understand the connections and the similarities and the unique features of RPT radiobiology, both the biological effective dose and the normal tissue complication probabilities, the acronyms of which you can see here. And so then I'll spend some time, time permitting, going over those, and then hopefully I'll be able to conclude by talking about some thoughts on the future outlook of this field. And so without further ado, let's get started. So dose distributions in RPT versus EBRT. So Carlos Uribe, who is of your group, and I are both members of this consortium, the RPT Tech, Radiopharmaceutical Therapy Tissue Effects in the Clinic. And so the motivation of that was in analogy with what they have in external beam, it's called Quantec. And in Quantec, they have for normal organs various dose limits for various normal organs at risk, dose limiting organs in external beam radiotherapy. And we don't really have dose limits like that are specific to radiopharmaceutical therapy. And so oftentimes what ends up happening, as you see in this letter to the editor that Carlos and Anna Kies and several others wrote, is that RPT clinical trials are often facing this artificial glass ceiling where we have dose limits derived from evidence in external beam radiotherapy that we are just artificially enforcing, and that kind of is setting a lot of these RPT agents up to fail and not really explore their full potential. And so, and very often it's observed that patients in RPT can handle doses up to this synthetic EBRT value, but for various reasons, RPT development is hindered because they have these EBRT dose limits that serve as a glass ceiling, even though we know, those who are in the community, that the dose limits from EBRT are not really transferable to or correlate well with toxicities in RPT. So I think the question really we want to ask is, why is that? Why do EBRT absorbed dose limits fail for RPT? And we kind of know kind of the general answer, the qualitative answer, then in a nutshell, is because the radiation biology of radiopharmaceutical therapy is different from external beam radiotherapy. There's just the fundamental nature of how radiation leads to various dose responses is not the same between the two modalities, and we don't fully understand those differences. But a good summary of the issue I've liked is from this MERD primer. Maximum tolerated doses for specific organs are not directly translatable from external beam to internal radionuclide treatment planning because of radiobiologic considerations related to temporal and spatial differences. That is, differences in dose rate / fractionation and non-uniform distribution between the two therapeutic modalities. So people kind of know what this is kind of qualitatively, but I think one of the things I'm really interested in is understanding the nature of these differences and exactly how big they are in a more rigorous, deeper, quantitative way. And so that's really what I'd like to spend some time talking about today. And to understand why the radiobiology is different, it all starts with the precursor, that is dosimetries. So I think external dosimetry is very different, as I'm sure you know, from internal dosimetry. And what it comes down to is the fact that again, the unique role of physiology and physiological pharmacokinetics. In external exposures, the physical absorbed dose is driven entirely by physics and by kind of, in the case of, say, a LINAC-based treatment, kind of things that we can control like beam angles, collimators, so on and so forth. In internal dosimetry, you have this special role of patient-specific physiological pharmacokinetics, and that's just not present in external beam. And so it leads to very different controlling factors that determine the dose and the dose rate distribution. And in a nutshell, what it comes down to is that we have very qualitatively different characteristic dose distributions between the two modalities, both at the cellular level, because, so you see, this is a figure taken from a review article by George Sgouros. You see at the cellular level, RPT, because it's controlled by these kind of physiological processes that are not directly under our control, it has this kind of more semi-stochastic and kind of heterogeneous cellular distribution, as opposed to, say, an external beam. You have a very coherent beam at the cellular level. But on the other hand, there's also the fact that if you zoom out and look at the whole organ level, this is the whole idea of see what you treat, treat what you see. The dose is really strongly correlated at larger scales with overall anatomic and physiological features in RPT because of the fact that, again, it's driven by physiology and anatomy. Whereas, again, in external beam, the characteristic large-scale dose distribution is driven by what the planner controls. Can we do some treatment planning? So it's very only weakly correlated with kind of anatomy and physiology. So a good analogy, it's not a perfect analogy, but it's one that I like to use in describing the differences between the doses, is that EBRT is kind of like a smooth, well-kept like golf course or set of hills, which is kind of, the grass is very, very clean and the steps, and there's very little difference from step to step. But then over a longer scale, it looks kind of like you have these hills and valleys and so on and so forth. Whereas RPT, locally, you have a lot more roughness and it's very unkempt and unmowed. And so there's a lot of unevenness in the grass. But then kind of on a larger scale, it kind of has this flat order. And so it's kind of more structured on a larger scale. So that's kind of the way I like to think about the qualitative differences between the two. And the issue is, as far as radiobiology goes, the bulk of our existing collective radiobiological knowledge, including the various concepts and particularly mathematical models and how we apply those in the clinic, those have been developed for EBRT, for doses that look like the left. And then RPT is another ballgame. And so then it's not at all clear, okay, we have various ideas on radiobiology. Which of those ideas are specific to EBRT and what ideas need to be adapted for RPT? Can we adapt them? What are the differences? And so that's really, I think, something that I'm very interested in, I think, which is a big unknown in the field. And so I think if we are to understand radiobiology of RPT, we must first understand EBRT radiobiology, so that which again, as I said, is what is more well developed. And if we understand that first, then we can kind of start to think about, okay, how is RPT different? And so with that in mind, I'd like to just take a few minutes to give a very, very brief and far from complete overview of external beam radiobiology. I'm basically going over like 2 months of my course in maybe like a few slides. So obviously I'm not going to be able to give the subject complete justice, but just a flavor of some of the most important elements of radiobiology in external beam radiotherapy, I'd like to just spend some time going over. Before I do that, are there any questions? All right. So when you're talking about radiobiology and external beam radiotherapy, or in more generally for any like dose response, I like to decompose the process into like two pieces. The question is, how does the radiation kill cells, like kind of on a local and comparably short time scales? And then kind of the somewhat longer time scale and larger organ level question is, how does the distribution of dead or damaged cells translate to an overall collective response? So for example, here you see the kidneys, that some fraction of the cells get killed or damaged, and then the patient presents with a follow-up response, creatinine clearance, GFR, real-time dynamics, so on and so forth. So kind of there's these kind of two modular pieces of the radiation dose response that I like to kind of compartmentalize and think about when analyzing radiobiology. And so let's go through them individually. How does radiation kill cells? And so the paradigmatic clinical model is the linear quadratic model, which basically says that you have a linear and a quadratic contribution to the survival probability on a log scale, of course, as a function of absorbed dose. And this is, again, for EBRT style, like acute single exposures, single fraction, very uniform, coherent, uniform quality beam. In such a case, we have a pretty good intuitive mechanistic interpretation of the parameters of the linear quadratic model. It's been shown through various supplementary experiments that you can interpret that alpha term as single track directly lethal damage, particularly specifically of the chromosomes and the DNA double strand breaks. And then the beta term, that double track term, that can be interpreted as individual tracks that are not lethal on their own, but can accumulate to kind of create collectively lethal damage. So you can imagine each track making a single strand break, and then together those combining to make effectively an overall double strand break that leads to lethality for the cell. And so that is kind of, at least for the very simple kinds of exposures, the form of the curve and how we can interpret the parameters of that model. And of course, in practice, real therapies in external beam are rarely given in a single fell swoop, but more commonly, of course, it is the idea of doing what's called fractionation. Although, of course, one of the big hot topics these days is flash radiotherapy, which is doing very, very high dose rates, all the dose in one fraction. And so maybe that paradigm might be slightly changing, but historically, most external beam treatment is done in kind of fractionated regimens where you deliver a certain amount of the dose one day, then the next day, and so on and so forth. So you distribute it over the sequence. And so the question is, okay, how do you adapt the cell survival curve and cell survival model accordingly? So let's work it out. So suppose the total dose D is delivered over, again, n uniform equal fractions, dose D over n for each little d, of course, representing the dose per fraction. Then what you can show is that you can kind of take each fraction individually and aggregate it, and then you get basically for the fractionated regimen, a survival curve that is often more easily written or conveniently expressed in terms of this derived quantity, which is called the biological effective dose, which is the total dose over the entire treatment multiplied by this parenthetical factor, which depends on little d, or the dose per fraction. And so that's a dose rate effect. And so you can imagine, if you take the continuous limit, if you imagine the limit of, suppose you had hypothetically infinitely many fractions, so you essentially, this comes to a situation where you are kind of like continuously giving the radiation over time so there's like not really any gaps between fractions, but it's just a steady stream of radiation. Then you could imagine in that limit, that BED kind of reduces directly to the total absorbed dose And so then it becomes a purely exponential dependence of cell survival. As we can see, that's kind of one extreme limit. But more generally, the idea is that we can relate different doses delivered with different fractionation regimens using this BED and this way of interconverting between fractions. So that's kind of the very basics of how radiation kills cells in external beam doses. And so now what's the second piece of that dose response? How robust is the organ to the loss of different cell types? So how does, okay, we've killed some cells, how does that translate to some response and some clinical endpoint? So it's very much dependent on the specifics of the organ, but in general, you can imagine a tissue or an organ as being composed of a number of compartments of cells with various different general categories of cell types, namely undifferentiated stem cells, partially differentiating cells, and fully differentiated functional cells. These are cells that don't really divide, but are basically like your muscle cells that are just fully differentiated and basically serve the function of enabling your muscles to work properly, or likewise your nerve cells, or so on and so forth. So you have these kind of complementary cell populations, and a particularly useful classification of various tissues is the Michalowski classification, which says that you can think of tissues or organs as being generally, roughly speaking, of two types: hierarchical or H type, or flexible F type. So when you have a hierarchical tissue, you can imagine something where there's always a stem cell niche in addition to the fully differentiated functional tissue, so that if there's ever damage to the functional tissue, that stem cell reserve can immediately replenish. So this is particularly apropos for something like, say, bone marrow or immune tissue. And on the other hand, you have an F-type tissue where there's not really, typically speaking, a stem cell niche, but if need be, cells can revert transiently to a stem cell-like state as needed to replenish. And so those, as you would imagine, have a longer recovery time than H-type. But this is kind of the general kind of picture of how these different types of cells and different compartments interplay to determine the functional response and how the organ responds to it. So at a very general level, you can kind of break down the time course of a dynamical response into this kind of like very, very crude qualitative picture. There's generally a time delay, which again varies depending on whether you have normal or H-type rather, or F-type tissues. And then you have kind of, depending on how many cells get incapacitated and or not incapacitated, you have a certain time scale of recovery that emerges from the kind of stem cell repopulation dynamics and how that translates to the transient time-dependent functional capacity of your tissue. So that's kind of like the general picture. And what emerges from this picture and what we've really come to understand is that it's not enough in general when you're talking about a whole tissue or an organ to just say that there's some average dose. It's important to understand where that dose is, because if you have these heterogeneous multiple compartments, the overall effect depends on which cells are going to get killed. And so this is what's called in radiobiology the volume effect, which is that the overall tolerance dose for an organ for a given toxicity endpoint depends on the specific fractional volume, in other words, where the organ is irradiated. So, for example, when you're talking about, say, how much functional capacity there is at any given time, then you can imagine even within those functional cells, each of them serving as a sort of what we call functional subunit, and then on aggregate, they work together to kind of determine the overall functional capacity of the organ, and which FSUs and how many of each FSU gets knocked out determine the magnitude of the acute toxicity. And then on the other hand, if you're interested in an endpoint like long-term recovery or long-term toxicity, then in addition to, again, the functional cells, you have to understand those stem cell compartments. You can kind of think of those as a separate quasi like FSU-like component, but in this case, they are interplaying to determine, again, whether or not there is long-term recovery or irreversible damage. And so then the question is, okay, you have these different compartments or FSUs or whatever you want to call them, and the interplay of them determines the overall toxicity endpoint. And so very roughly speaking, we can imagine for a given tissue or an organ, various FSUs or compartments as being kind of falling into one of two extreme rough paradigmatic limits of kind of like toy models of functional organization. And these are, again, extremes, but I think they're good approximations for some cases. The one extreme is the serial organization, which is where you can imagine a situation where all the different FSUs or components are extremely interdependent, so that if you knock out even one or even a few of the pieces, the whole organ gets incapacitated. And so a classic example of that is the spinal cord, because again, it's linked in this kind of serial chain. And so you knock out one neuron in the nerve fiber that kind of incapacitates the entire nervous system, or spinal cord at least. And then on the other hand, on another extreme, you have something that's called a parallel level of organization, which is where each FSU or compartment kind of acts autonomously and independently of the others. And so basically, when you ask the question of, okay, how much the total organ gets incapacitated, that depends on how each of the individual pieces get incapacitated. So it's proportional to what fraction of the individual FSUs remain in your reserve. And so, again, these are extreme, very stylized limits, but they're good approximations for certain organs. Not always, but sometimes they're good starting points. But in general, though, most organs are more realistically mixed combinations of them. So, for example, in the kidney, you have an example of the parallel serial model. This is a classic paper in '92, which showed that, okay, you can take the nephron of a kidney and you can kind of decompose it into a number of different FSUs, and parts of that circuit, if you will, have parallel elements and other parts have serial elements. And so you really have to kind of understand how the combination of both serial and parallel organization interplays to determine overall organization and functional robustness to knock out of this, that, or the other FSU or compartment or what have you. So I think in this '92 paper, one of the models they proposed to describe this more general realistic kind of a combination of serial and parallel components, you can kind of break down the overall organ toxicity or endpoint into these two pieces. The first part being kind of a Poissonian survival as a function of dose. And this is basically related to the magnitude of, again, those individual alpha, beta, LQ parameters for that specific, the cells in that specific FSU. And then that second part, which is this volume effect, kind of asks us, okay, how, when they get knocked out individually, how does that translate to aggregate NTCP? And you have this new ingredient, which is called S, the seriality parameter. And it kind of measures kind of the fraction of components that are serial as opposed to parallel. So if you have something like a highly serial organ, S equals almost 1, it can be shown that the NTCP is kind of limited and controlled by kind of the maximum dose to an FSU in the organ. And so that's why, again, for spinal cords, you're limited by your hotspots. And then for the other extreme parallel organs, it reduces to kind of coming down to, again, the mean dose. And then more generally for like in between, this is why for some organs, they treat not necessarily the mean, not necessarily the maximum, but maybe an intermediate, like a dose volume histogram metric, something in between the mean or the max. It's because depending on the organ, there might be a slightly different nature of that volume effect. So that's really where the DVH-based guidelines in EBRT come from. So that's kind of a whirlwind tour of kind of like the basic elements of EBRT radiobiology. And any quick questions?
In
All right,
practice,
so,
Tahir, if I may
go ahead.
ask, in practice, how much are these models, especially towards the end, these parallel versus serial models, in practice, how much are they actually used?
Well, it's okay. So yeah, no, not maybe explicitly in the clinic per se. So I think, again, usually in EBRT, again, people don't consciously think about the radiobiological model as they're doing the treatment planning. Most EBRT planners, again, they do dose volume histograms and they're just thinking about optimizing some dose constraint for this organ, that, or the other. But the roots of it, again, are based on models like these. And so you can imagine them, they're not used directly, but like the reason that we do planning based on certain physical dosimetric objectives is because of the underlying radiobiological foundations. And so does that kind of make sense?
Yes. Yeah.
Sure. All right. And so, okay. So that's kind of how radiobiology, and again, I think so kind of that's a good way of understanding, okay, there's these radiobiological foundations behind the various reasons they do make decisions that they do in external beam radiotherapy. But again, the question is, okay, how is RPT different? And so again, I've told you about these two bits of the dose response. And basically what I'd like to spend some time talking about is some of the work I've been doing on trying to kind of understand how to adapt, or like how RPT, how those pieces have to be adapted differently for radiopharmaceutical therapy. And so the first part, of course, is how does radiation kill cells, that BED, efforts towards a more universal way of understanding the biological effective dose in cell kill. So if you take our derivation, so if you recall, I showed you this very, very rough derivation of how we calculate the biological effective dose and how it can be used to relate different fractionation regimens in external beam. But when I did that derivation, I made a number of implicit naive assumptions that I didn't tell you about. And these are not the only ones, but some of the important ones that I made were that there's no repopulation in between the fractions, and that there's complete repair of any sublethal damage in between the fractions, and that every cell receives the exact same dose, exact same radiation quality, is very coherent. And I did that when I assumed that basically each fraction, you could just multiply them independently. And so you had essentially the same alpha beta terms at each fraction. You can just kind of multiply them individually. And in other words, there wasn't really much memory in between. Basically, fraction 2 didn't have much memory of fraction 1. That's why you could multiply their individual contributions like this. In general, though, these approximations sometimes hold, but in general, they're never universally held in either RPT or external beam radiotherapy. Now, it's probably fair to say that they hold less so in RPT than EBRT most of the time. But even in EBRT, they are interested sometimes in trying to go beyond this, and it can be important even in EBRT. Even though it's probably more important in RPT, it's still important sometimes in EBRT. And in fact, people have tried to go beyond a naive BED, and there are a number of various corrections that people have derived, some of which have been adapted to the RPT realm. So these are some slides adapted from Rob Hobbs' APM Summer School slides showing some of the most common non-naive extensions that have found applications. So in particular, when you have something like a pharmacokinetic dose rate with a MERD calculated dose, and then you time integrate it to get the absorbed dose. If you want to have a better understanding of dose rate effects that includes sublethal damage, incomplete repair, you have what's called a Leah-Catchicide factor, which accounts for incomplete repair between fractions or between cycles. And if you allow for, say, repopulation in between cycles, you can have an additional correction factor to the survival fraction, as taken from a paper by Lydia Strigari. And so on the bottom right, you have a slightly less naive formula for BED that, again, originally these corrections were developed in EBRT, but they can be partially adapted sometimes to be a bit more useful for and make RPT make the BED, shall we say, a little more useful for RPT some of the time. But that said, in general, though, there's still no universal formula for the BED that applies to an arbitrary, general, clinically relevant RPT exposure. And I think this schematic kind of gives a good breakdown of the problem. So again, really, again, what it comes down to is, again, EBRT is a very uniform, very coherent, very clean dose at the cellular level. RPT is much rougher and messier. And so, and in particular, suppose you had something like a mixed combination. Suppose you wanted to treat a patient with a combination of alpha emitter, beta emitter. And so then you have lots of different doses with different agents, rather, with different pharmacokinetics, different radiation qualities. We don't really have a good clean formula for the BED if you have kind of a very complex mixture of different types of radiation, each with slightly different pharmacokinetics. And that's really what you would need if you wanted to do something like, say, understand the biological equivalence of combination RPTs, if you want to try out different synchronous versus asynchronous cycle delivery, so on and so forth. You really need to have a way to understand BED for these kind of more general, messy, heterogeneous exposures. And we don't really have a parsimonious good model for doing that. Now, on the other hand, there is work in what's called mechanistic modeling and mechanistic simulations. And that's things like very detailed track structure calculations. So for example, things like Topaz and Bio or GANT4DNA, although of course those are developed primarily for EBRT, but some of the radiobiological models can be adapted to be simulated for RPT style exposures. And then you have kind of slightly more reduced, but still mechanistic reduced order models, things like Steve McNihan developed this thing called MEDROS, Mechanistic Model of DNA Repair and Survival. So again, these kind of are based on the more fundamental physics, chemistry of the DNA damage, the repair process, the cell cycle death, so on and so forth. So maybe if we had something like that, that might in principle allow us to understand kind of an arbitrary, complex, heterogeneous RPT exposure. And so that would be a great advantage. But on the other hand, it comes with a lot of problems that, well, I shouldn't say problems, shall we say, current challenges or disadvantages, which is that they're computationally expensive, but more importantly, in my opinion, there's a lot of uncertainties because they're very complex. There's a lot of uncertainty in what's the right mechanistic models to select, how are we going to get the parameters, so on and so forth. And so that computational expense bottleneck, that's, in my opinion, not as important. I mean, it's there, but I think there's strategies in place for addressing it. Things like GPUs, hardware advancements, and then some of the work I've done in collaboration with some colleagues at Georgia Tech was developing kind of these accelerated lookup table track structure calculations to kind of basically use these lookup libraries to accelerate these brute force Monte Carlo calculations. And so we just published this paper in Frontiers in Nuclear Medicine. We were able to replicate some in vitro results on DNA damage and repair for lutetium-177. And we were able to do it with a 4 orders of magnitude speed up compared to the brute force calculation. So this is just one example, but then, yes, there are definitely ways to kind of speed up these calculations, and it's still a work in progress, but I think that, in my opinion, is not really the major long-term obstacle. In my opinion, the much more important long-term problem is the overabundance of empirically unidentifiable model parameters. Because again, if you add more pathways, more kinetics, more potential etiological mechanisms, you add more model parameters. And even the simplest of these mechanistic models can have at least 10 parameters. And it's a tall order to reliably estimate them. I mean, we talk about patient-specific parameters, let alone, it's hard enough even to get population level. I don't think you can even really get reliable population level parameters for something like with 10 or 15 parameters. Forget about patient-specific. And in part, this is actually part of the reason that when we can use LQ style models, like in EBRT style exposures, that's really, I think, the reason it's so clinically successful, because it's rigorous radiobiologically, but it's also parsimonious enough that you can reliably estimate the parameters, and people are confident enough to take those estimates. And so we really need some way to kind of work around this parameter unidentifiability problem. And so what I've been working on with a colleague of mine from BYU, Mark Transtrom, he's a biophysicist who developed, originally in the context of systems biology, an approach to these kind of large-scale systems biology models. How to reduce them is called the manifold boundary approximation method. And it's a beautiful paper and it's a beautiful method, but basically, in a nutshell, it gives a way to kind of create controlled approximations to kind of large, many parameter models, so that we can kind of take something mechanistic and reduce it into something that is still mechanistic, but capable of actually being estimated with realistic data sets. And that's kind of what I've been working on doing with applying the MBAM to these high parameter, large multidimensional parameter spaces in radiobiological models. And so as a proof of concept application, we started by applying it to a kind of that MedDRES. And so again, this is not as detailed as the radiobiological track structure mechanistic models, but it's still mechanistic, and it's still got like 13, 15 parameters. And so it's still somewhat messy as far as whether we can actually estimate it in general for patients on a patient-specific or even a population level basis. And what we showed using MBAM was you can take, this is what it looked like on the before, and then afterwards you reduce it to a 3-parameter model, which has essentially indistinguishable predictions. And so this was a proof of concept application. Again, this was just meant to show that we could do it, but work is currently in progress. We really want to kind of generalize and adapt it to kind of the really more detailed mechanistic models that are really more appropriate for, again, as I said, these combination RPT exposures, very heterogeneous, lots of stochasticity, lots of variable LET distributions. And for those, I think we need even more detailed mechanistic models. And so that is work in progress. So that is what I've been thinking about as far as that first part of the dose response, the BED. How can we kind of adapt it or generalize it to RPT? Now, what about that second part? Okay, NTCP, volume effects. So volume effects in RPT are hard. And again, I think a good summary of the problem is in this a paper by I think Lydia Strigari and colleagues, which is while partial kidney irradiation in EBRT usually implies the irradiation of a fraction of hundreds of thousands of nephrons of the kidney, in RPT, it implies a differential irradiation of a whole collective, blah, 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 which implies evaluating the assumption of parallel tissue organization throughout kidney tissues. So that's a mouthful, but the translation, or like my read on it anyways, is that to first order, for the kinds of doses you see in external beam radiotherapy, the kidneys and several other organs, they appear parallel, and to a good approximation, they are parallel, but at a more fundamental level, they're really not. So for example, if you take, this is kind of a stylized circuit model for like the kidney nephrons, and because of the way that the doses in EBRT are, and that they kind of are localized very strongly to kind of that periphery, to a good approximation, only like that periphery matters. And so you can see what it looks like in EBRT. It's as if only that parallel single component really comes into play as far. And so for the purposes of dose toxicity modeling of the kinds of doses seen in EBRT, you can almost neglect all those other parts and just assume there's kind of like this parallel organization. But in general, and especially for like these physiology mediated doses you see in RPT, you cannot do that. And so this has to be re-examined. And so then, okay, so what are we going to do? So again, now I said that even it's a good approximation. I said in EBRT, you can make the approximation that kidneys are parallel. And it's good enough because if it weren't good enough, we wouldn't be able to do treatment planning at all. But even in EBRT, they know that they can do better. And so they had the Quantec papers in 2010. And then since then, especially with the rise of machine learning, people are very interested in the external beam community in seeing if we can discover more accurate, high precision correlates of toxicity, even again, for external beam. So this is a nice review from PMB on spatial descriptions of radiotherapy dose. And I think the really big thing that I think is very exciting is that such approaches are culminating in the application of computationally intensive processes such as machine learning and the application of neural networks. And so there, the idea is, okay, they know they have Quantec guidelines, very approximate models for various organs, but they know that there's more to be learned and they can do even better. And so in EBRT, they're doing this, trying to discover these higher order associations with machine learning. There, okay, it's a useful improvement. It's valuable. It'll help. It'll make the improvements, the treatments better. But I mean, treatment planning has been possible before then. It'll be possible even if we don't get there very quickly. On the other hand, for something like RPT, understanding kind of these kind of higher order corrections due to these kind of more unbiased models of the volume effects, that could be the difference between whether you predict a toxicity to be dose limiting or not. So it can be kind of like a much bigger deal as far as like whether treatment planning or treatment is even possible. So I think it's worth kind of seeing for our purposes in RPT anyways, maybe there's these higher order effects that might be useful to EBRT, but might be really, really important for understanding how to adapt volume effect modeling for RPT. And so I basically, what I did was I developed a theoretical proposal, which I published a few years ago, was showing that, okay, they're using machine learning and neural networks to discover these kind of higher order correlates of dose toxicity. And so what we showed was that some of those neural networks could be interpreted as kind of like higher order relative seriality models. So I explained that relative seriality dose response model. It shows that it can be shown that it can be interpreted as kind of a very simple neural network. And so then you can imagine if you have a deeper neural network with more layers, one way of kind of making sense of that is that it's discovering kind of higher layers of features in the functional organization. So that maybe if you have a little bit of data, for EBRT, for example, and you train it on a kidney, their own neural network might learn that it's a parallel organ, which is again what's in Quantec. But as you feed it more data, it might discover these higher order corrections due to, again, these deeper layers of functional organization. And those might be especially important, again, if you want to do something like RPT volume effect modeling. And so that's kind of the theoretical proposal. And so one of the yes.
Tahir, is capital N, that's a number of, I guess, functional subunits or something? How would you set that in these, like you go D1 to Dn, how would you set the number of nodes?
That's a good question. So yes, I'll try to explain that through an example. So I mean, that depends on the organ. Again, the pilot application that we've been trying for is the salivary glands. And so salivary glands, they're a common organ at risk in both head and neck external beam radiotherapy and, among other things, PSMA radiotherapies. And again, just like for kidneys, we have kind of these mean dose guidelines for parotid glands that are okay for EBRT, but they know they can do better. And so this is a paper in the Red Journal from the external beam community showing that even then they know that salivary glands present both parallel and serial components to the function of the glands with a complex spatial pattern. And so, okay. So now we know that, okay, it's especially important for us in RPT to understand this problem, just like for the kidneys, because we really can't assume that it's parallel in general for the types of doses that we might see in RPT. And so what I've been trying to do with a student, Chun-Ming Gu, is to kind of see if we can implement the DRSN to the salivary gland. So in the case of the salivary gland, in this very initial stylized model, so then you ask the question, okay, how do you decide on the level of, shall we say, functional organization or functional compartments? And so here what we have is, okay, say you have 4 salivary glands. At a very simple level, you can imagine the salivary gland as being composed of kind of, at a lumped element level, kind of an aggregate acinar bulk, and then kind of like this collecting duct network. And so then, if you wanted to go deeper into that, then you could imagine that duct network as being decomposed ever further into kind of macroducts, mesoducts, microducts. So answering your question, Armand, in the case of a salivary gland, you define it in terms of, say, you define like your FSUs in terms of, okay, the ductal network and the acinar tree. And then again, I guess more generally, I guess for different organs, I guess you'd have to take it on a case-by-case basis. But I guess the idea is you'd have to look at kind of the anatomy specific to that organ, and the macro anatomy and the micro anatomy, and kind of have kind of an understanding. It's not really a hard rule. It's kind of more of an art form at this point. And but I think, at least in the example of the salivary glands, this is how we've been trying to implement it. And we have some encouraging preliminary results showing that compared to mean dose classifiers, the DRSN shows improved training and, more significantly, improved generalization. Because again, remember, what we're really interested in is models that generalize to unseen data. Because for RPT, that's what we really need. And so again, these are still, however, I should caution, very preliminary, and we need to really undergo a more comprehensive analysis. And in particular, what we really need is a more comprehensive uncertainty quantification before we can conclude anything more solid. But we believe the results are encouraging. And so whether or not they'll hold up, or whether they'll be able to be done more generally for other organs, we don't know. But it's something that we're excited about. So anyways, okay, so that's basically my work. I guess I could pause for questions, but I'm almost done anyway, so I'll just finish and then take questions. So, okay, so I've told you all about radiobiology and EBRT, how it's different from RPT. So, okay, so now how does this tie into kind of the bigger picture? And so, as Arman mentioned at the beginning, I've been working with Arman and Carlos and a bunch of others on computational nuclear oncology, which is kind of our vision that we have this set of mechanistic principles that guide the prediction of dose and dose response in RPT. And so radiation biology, which is again the translation of the physical dose to the biological dose and outcome, that's kind of the downstream part of that. But then you also have that upstream part, which is again what you are all very good at, the physiologically based radiopharmacokinetics, which again you have to estimate from nuclear medicine imaging, so on and so forth. But combining that with radiation biology, we get this kind of emergent field of computational nuclear oncology that I believe is the future of RPT. And so as we make advances in our basic and applied understanding of these scientific principles, those are going to eventually make the foundation for the future of how we do personalized RPT, in our opinion. And I think there's many benefits to that. And so this diagram is taken from, it was created actually by Carlos for a paper that we just submitted. We have this kind of vision of using these multi-scale CNO framework to go beyond one-size-fits-all RPT to do kind of personalized dosimetry and RPT-specific treatment planning. And even like, though I think we're still got a lot more work to do. I mean, these are some good, interesting preliminary results with, well, not, I shouldn't say preliminary, but some initial results from the Dososphere trial showing that even though I guess yttrium-90 is maybe, depending on who you talk to, not exactly a radiopharmaceutical therapy, but still it's somewhat close. Even there, okay, these are just encouraging results showing that there are real important benefits to doing personalization. And so we really believe that as time goes on, we are going to kind of bring CNO into the framework and to kind of more generally move towards a precision RPT paradigm. And so we hope to see a lot more results like this in the future as CNO gets developed. And with that, both the radiobiology and the pharmacokinetics in concert being developed and working together to enable things like this, and to enable more exciting things, even more exciting things. Like, so for example, say you want to do treatment planning for not just a mono RPT, like just a beta RPT or a single alpha RPT, but suppose you want to do something like a mixture of alphas and betas, or you want to design a cocktail of different radiopharmaceuticals, again, each with various different pharmacokinetics, different propensities for this and that or the other. Then as we get developed, for example, as I talked in one part of my talk, as you develop a deeper understanding of how to understand biological effective dose for such arbitrary combinations, that will kind of be applied directly to make things like this a more mainstream. And then if we understand better the relationship between volume effects in EBRT versus RPT, as I talked about in the other part, then it might enable more routine combination therapies for, say, using external beam to treat kind of primary local sites, and using radiopharmaceutical therapy to treat more disseminated metastatic disease, but using the same level of treatment planning rigor on both. And so, I mean, right now, I think it's currently doing these kinds of things on a routine basis in the clinic. It's currently a blue skies daydream, but I do believe that we will get there, and computational nuclear oncology will be an indispensable part of it, in my opinion, and in the opinion, I'm sure, of Arman and Carlos and many of you. And so, in summary, so the radiation biology of RPT, I hope I've convinced you that it's different from EBRT, and it originates again from dosimetry. And because of that, there's kind of systematic differences that need to be understood better. And there's still a lot of work to be done in understanding that radiobiology. And I hope I've excited you with some of the work that I showed on trying to address some of those differences. although I didn't cover all of them, I hope that it whetted your appetite for more. Because I do believe, as I've mentioned just now, that advances in our radiobiological understanding will have long-term implications for computational nuclear oncology and for the future of dosimetry-driven treatment planning in radiopharmaceutical therapy. And so with that, I would like to acknowledge my students and colleagues and collaborators, both at Johns Hopkins and of course externally, including Carlos and Armand, of course, and several other, particularly people on the SNMI AI task force and some of my funding sources. And with that, I thank you for the invitation. I'm happy to take questions.